Hello friends, here I would like to explain lac operon. So, lac operon is an operon which help for the catabolism or breakdown of lactose. It is an operon which help for the breakdown of lactose into two. One is called glucose and second one is called galactose. This is the main function of lac operon. So lac operon is an operon which help for the catabolism or breakdown of glucose into, which help for the catabolism of lactose, not glucose, which help for the catabolism of lactose into glucose and galactose. And the resultant glucose can be used for the cell to produce energy through glycolysis. And this resultant product also, galactose, can also used by the cell for producing energy through another operon. That operon is called gal operon, which will be discussed in next class. Okay. So the main function is to cleave or break or catabolize lactose into glucose and galactose for the purpose of producing energy. Okay, that is called the lac operon. Okay. Now coming to the structure of lac operon, it consisting of three structural genes, namely lac Z, lac Y and lac A. Okay. This lac Z gene produces an enzyme. That enzyme is called beta-galactosidase. So lac Z produces an enzyme called beta-galactosidase. Like that, lac Y produces a protein which is found on the cell membrane of bacteria. Okay. That protein is called, lac Y producing protein is called galactoside permease. Beta-galactoside permease. It is a protein found on the cell membrane of bacteria. Okay, its function will be discussed later. Okay. Third type of structural gene is called lac A. It produces another enzyme. That enzyme is called transacetylase. It produces another enzyme called transacetylase. Okay. These are the three structural genes present in the lac operon. Okay. Now coming to the function of this gene product. Coming to the function of this gene product. First of all, beta-galactosidase. This beta-galactosidase perform mainly two functions. First function of beta-galactosidase is to convert a disaccharide called lactose into glucose. Okay. And glucose and galactose. This is the function of lactose. It breaks galactose into glucose and galactose. That is the main function of beta-galactosidase gene product called beta-galactosidase. Not lac Z gene product called beta-galactosidase. Function is to convert lactose into glucose and galactose. Its second function. It also has second function. Okay. Its second function is it can also able to convert lactose into another compound called allolactose. Lactose into another compound called allolactose. Okay. So these are the two main functions performed by this beta-galactosidase. Okay. And second one is called galactoside permease. Permease means an agent or protein which can able to permeate. Okay. So the function of permease is to transport or allow. Actually, permease is present on the cell membrane of bacteria. That is, lac operon carrying bacteria. Okay. So permease is usually found on the cell membrane of lac operon carrying bacteria. Okay. And it allow or it transport lactose present in the external medium of bacteria into the bacterial cell. So permease function is to transport lactose from outside towards inside the bacterial cell. Okay, that is the function of permease. Then coming to the third enzyme called transacetylase, its function is not clearly discovered. Okay. But it has some role in transporting lactose from external medium into the internal medium, like that of permease. But they have a specific proof for that. That is why I am saying its function is not yet discovered. Okay. Now, this lac operon consisting of a common operator and a common promoter. Okay. Here, this operator acts as the function of operator is to act as the binding site for a regulatory protein. To act as the binding site for a regulatory protein. So that operator is called, the operator present in the lac operon is called lac O. O means operator, lac for lactose. Okay. So here the regulatory, this lac operon carrying operator act as the binding site for a specific protein called the regulatory protein. Okay. Here, this regulatory protein is an active repressor. This regulatory protein is an active repressor. So, lac O, the operator site of lac operon, act as the binding site for a repressor regulatory protein, which means repressor is a regulatory protein which acts as a repressor or inhibitor of lac operon working. Okay. And coming to the structure of this regulatory protein has two binding domain, two binding site. One is called DNA binding site. One is called DNA binding site. One is called DNA binding site. Okay. Second one is called, this is called second site. This is called substrate binding site. One is called DNA binding site. And second one is called substrate binding site. So, regulatory protein called the repressor consisting of DNA binding site and substrate binding site. By using its DNA binding site, it always bound on the lac operator. By using its DNA binding site, it always bound on the lac operator site. Okay, and it also consisting of a substrate binding site. Okay, here the substrate is lac. Here the substrate is an allolactose. So here allolactose act as the substrate of this repressor. When allolactose bind on the substrate binding site, it results in the modification of DNA binding site. Okay. If DNA binding site modify, then the repressor protein cannot be able to bind on the operator. At that instant, lac operon start to work. But when allolactose is not found on the substrate binding site, its DNA binding site will be like this, active. So it can be able to bind on the operator and repress or inhibit the working of lac operon. Okay. And this regulatory protein are encoded by a specific monocystonic gene, which means a gene consisting of only one structural gene and which is encoded by a common promoter. So it's an example for a monocystonic. Monocystonic means a gene consisting of one structural gene and one promoter. Whereas all operons like this lac, all operon are example for polysystonic gene. Polysystonic means one promoter encode more than one structural gene. Okay, so here this gene consisting of this regulatory protein producing repressor regulatory protein gene consisting of one structural gene. That structural gene is named as LAC-I, and it has one promoter. That promoter is called the promoter of I, that is called LAC-PI. PI means the promoter of I. Okay, this is the broad structure of LAC operon. Okay, and this LAC operon is always off. This LAC operon is always off inside the bacterial cell due to the presence of an active repressor bound on the operator. So LAC operon is off, always inside the cell due to the presence of a repressor present on it. Okay, so this LAC operon is an example for negatively means negative regulation, negatively inducible operon. So this operon is an example for negatively inducible operon, which means an operon which is always off, inducible and always off due to the presence of a negative factor called the repressor on the promoter. Okay, actually operons are four types: negatively inducible operon, positively inducible operon, negatively repressible operon, positively repressible operon. But here this LAC operon is an example for negatively inducible operon. Okay, all these four types of operon are already discussed in the previous video class. You can view. Okay, so LAC operon is an example for negatively inducible operon. Okay, this is the broad concept of LAC operon. Now I am going to explain the regulation of operons. When this operon gets start to work, when this operon stop its working. Okay, that two processes are going to explain now. Okay. So when lactose is present, when lactose present in the external medium of bacteria, then the bacteria need to catabolize that lactose. For that purpose, LAC operon must start its working. Then only lactose can be cleaved into glucose and galactose. So when lactose is present, bacteria need to start the working of LAC operon. Okay. But when lactose is absent, then the bacteria no need to start its working. Okay. Then that process it can off. Okay. So now we must know how the working of LAC operon start in presence of lactose and how LAC operon stop its working in the absence of lactose. Okay, that will be going to discuss now. Okay. First of all, I am going to discuss when lactose is absent. Okay, so lactose absent. When lactose is absent, So when lactose is absent, repressor protein, I already told you, repressor protein has DNA binding site and substrate binding site. So when lactose is absent, definitely it bound on the LAC protein. then lac operon is off. That is the condition. Okay. Because the repressor is an active form. Here allolactose is not present. That is, it can able to bind on the operator. So when lactose is absent, definitely it binds on the lac operator thoroughly. So the operon is off because RNA polymerase bound on the lac promoter cannot move into the structural gene due to the presence of a repressor on the lac O. Okay, so lac operon is off. That is the condition when lactose is absent. But one more thing to understand here we have to understand a very important thing. I already told you that when lactose is absent, its working is off, which means when lactose is absent inside the medium, 98% time this operon is off. Not 100% of the time. When lactose is absent, 98% of the time this operon is off, which indicates that 2% of the time this operon is on. How this operon gets start or on in 2% of the time? The answer is when lactose is absent. Look, this is repressor. When lactose is absent, repressor is bound on it. Then this operon will never work. But at some specific time, this operon detaches from the operator site. But this detaching condition never exists for a long time. It again rebound on it for a long time. And after a long time, it again detaches. And it again rebound on it. So, I already told you that when the absence of lactose, repressor bound on the operator site, but this bounding is not a stable bounding. At some instant, it will detach from the operator. But this detachment never exists for a long time. This detachment exists for a short time. Again, it bound on it like operator. But at some specific moment, it will detach from the lac operator. At that side, RNA polymerase can be able to move through it and produces very low amount of beta-galactosidase, permease, and transacetylase. So, we must understand when lactose is absent, 99% of the time lac operon is off, whereas 99 or 98% of the lac operon is off, whereas 2% of the time it's in an on condition, working condition, and that very short time it produces very low amount of beta-galactosidase, permease, and transacetylase. So we must understand that when lactose is absent, lac operon containing bacteria produces very small amount of beta-galactosidase, permease, and transacetylase. Okay, but when we say that it is a negligible time, 2% of the time is a negligible time, whereas 98% of the time is a major time. That is why we are neglecting the 2% of the time. That is why we are saying that when lactose is absent, the operon is off. Okay. So, now I am going to explain what happens when lactose is present inside the bacterial medium. Okay. So, when lactose is present, okay, I am modifying when lactose is present. When lactose is present, this lactose, when lactose is present in the medium, Very small amount of beta-galactosidase permeases enters as it is already present inside the bacteria. So this beta-galactosidase transport the lactose present inside the bacterial medium into the cell. So when lactose is present in the external medium, this is lactose. When lactose is present in the external medium, small amount of permease transport this lactose into the cell through permease that is produced in 2% time. Okay. And when lactose reaches inside the cell, small amount of beta-galactosidase present in the bacterial cell convert it into allolactose, and it also convert it into glucose and galactose. And this lactose is converted into allolactose. This lactose is converted into allolactose. It also converted into glucose and galactose. Glucose and galactose by the cell. Okay. So this newly entered lactose has two fates. Some lactose are converted into allolactose by this enzyme beta-galactosidase. Some lactose are converted into glucose and galactose by this enzyme beta-galactosidase. Both functions are performed by this enzyme beta-galactosidase. And this newly formed allolactose then directly bind on the substrate binding site. Directly bind to the substrate binding site. Okay. Actually, this regulator protein is bound on it in an off condition. Okay. This allolactose bind on the substrate binding site. Okay. When allolactose bound on the substrate binding site, its DNA binding domain gets modified. That modification leads to the detachment of repressor protein from the operator. When this repressor protein detaches from the operator, then the RNA polymerase present on the promoter can easily move into the structural gene and transcribe all the proteins and enzymes. Okay. Then that condition is called the working of operon. Now the operon is working and produces more and more amount of beta-galactosidase, permease and transacetylase. That is why lactose can be cleaved into glucose and galactose, and after that the cell can able to produce immense amount of energy from glucose through glycolysis and galactose through another operon called galloperon. Okay, that is the main concept of lac operon. Okay, this is the broad concept of lac operon. Okay. If you like this video, please subscribe. Okay, thank you.